Is it okay now?
Yes, sir.
Yeah. Yes, sir. Cool.
So before I go into the talk, I have some disclaimers to make as well as some housekeeping kind of things. I am employed by Aventar as Senior Director of R&D, Innovation, Cell and Gene Therapy, Biopharma and Production. But this work that I'm going to present today is a summary of a lot of the work that I have done in my previous positions, and all the information is public. So it is none of the, and it is not done, and this particular work is not done at Aventar. So with that, I think regarding the, first of all, I have some, before I forget, few thanks to people. Thanks for all of you for joining, sparing your family time on a Saturday and the weekend. And then to Professor Redana for the opportunity, and to Deepika and Srinivas for facilitating a lot of things that are required in this kind of requirements. With that, what I'm going to do is cover a little bit of basics for CAR T therapy. And I'm not a clinician by myself, but I want to make sure that everyone understands this. So I have kept the talk to a very basic minimal science as much as possible. For hardcore immunologists who are on the audience, it might look like a very flaky. So please bear with me for the simplicity. So what I'm going to do the introduction is, what is CAR T cell therapy and how does the treatment process takes place? What are the advanced incidents associated with this CAR T therapy? And would there be an allogenic model approach for this? Which means the current CAR T therapies are all autologous. For those of you, autologous is cells taken from one individual, treat them outside, and then put them back in the same patient. So there is no graft rejection. There is no any kind of issues associated with immunology. On the other hand, allogenic is a model in which from a healthy donor cells are taken, then they are engineered or expanded or exposed to cytokines, and then put them back in different patients for treatment conditions. Each of them have their own advantages and disadvantages. We will go through. And what are the main manufacturing and challenges that are associated with therapy? And currently, CAR T therapy is roughly about $450,000 in US for one patient for one treatment. So by that, followed by that, we'll do some conclusions. So that if you look at the treatment approaches, each cancer is different. Cancer, even though is one kind of disease, all associated with the cell division, but it exists in many forms. And for the current research so far, for some of the cancers, hormone therapy is used. And if it is a solid tumor, ectopic surgery is used. And if it is blood related or bone marrow related, so it is bone marrow transplantation is done. And in all of these conditions, chemotherapy is done. And in few cases, there is, for example, breast cancer and all that, there is targeted therapy with the HEP2 antibodies, defining antibodies. And in few other cases, radiation therapy works for this. But let's hope all of them work for many of them. But there are other cancers that are recalcitrant. What I mean by recalcitrant is they recurringly occur, and none of these treatments work there. So that's where the immunotherapy comes into picture that has developed recently over the last decade. So that's why the importance of immunotherapy. When everything else fails, it is the only option that is left currently. So before I go into CAR T, I want to just get everyone on the baseline of what is that our immune system to respond with. So let's take, for example, if one is exposed to a virus or some other carcinogen. So what happens in the human body, the immunology kind of thing that I'm speaking about is what is called as innate immunity will kick in. So innate immunity that shows this blue line that is there, it goes up a peak and then drops back by 10 to 14 days time. And what is happening in that case is, for example, you must have heard from doctors, oh, it is a viral fever, you just don't take any medicines, take rest and fluids, you'll get away with. And that's where innate immunity is working. And this innate immunity is facilitated by first-line defensors, which are like natural killer cells, dendritic cells, macrophages, and then complement, neutrophils, eosinophils, all this kind of cell types. But while that is taking place, let's say if it is more than virus, and if it is a serious carcinogen that is doing it, the what is called as adaptive immunity also kicks in. And in this kind of adaptive immunity, the real T cells, the real immune cells, and B cells, they get into the picture. And what is not able to resolve by innate immunity will be resolved by adaptive immunity. So which means the T cell, this is called adaptive immunity because over the time, it has to take place. It will not take place soon after you are exposed. So therefore, the innate and adaptive immunity systems, both of them, they have to work together to tackle the virus or the carcinogens. And once this rhythm is disrupted, it can lead to quite a bit of viral spread. Viral will dominate. There will be hyperinflammation in the human body, leading to a kind of either cancer, pre-cancer, some of this kind of condition. So this is what is happening in those days when the T cells, look at this, when the T cells also drop off, when they resolve the tumor or virus infection, they are not touching the bottom zero, right? They are leaving a little bit of gap there, which means there are memory cells. So to bring you back an example, we all have taken COVID infection, COVID antibodies, COVID treatments and vaccines. So what it does is it resolves the infection. Not only the viral infection is resolved, a kind of T cell memory is retained. So the reason for that is when next time, when over a year time or 3-year time, if you are exposed to the same antigen again, so there is a similar response like this at that point of time. So that is the why the T memory cells are very, very important in that manner. So CAR T cell, what is CAR T cell? So the T cells are lymphocytes. Most of you might know that. This is a type of white blood cell. Now, when you go to hospital, they'll the WBC count and all that. So T cells are one of those white blood cell types that fight infection, and it is a part of the immune system. And what is a CAR? CAR is a chimeric antigen receptor. It's a kind of new protein. So most of these T cells have receptors, TCR, they are called TCRs. The ends of these receptors can be engineered, and you bring a chimeric antigen receptor. The name came because basically the red part that is shown is being synthesized completely, and that is being synthesized in tune with an antigen that is present on the tumor. So, therefore, since you have to engineer this particular receptor part, and this is a chimeric basically because chimera actually means, in literal terms, you have a human body, you have a human head, human body, goat body, and then a tail, something like that. It's a mixture of an organism. In reality, it doesn't exist. So therefore, it is synthesized. So this receptor is clearly synthesized to suit and to bind to that specific tumor antigen. So therefore, CAR T cell therapy is a gene therapy, not just a simple cell therapy. It is a gene therapy because the genes in the patient or T cells have to be reprogrammed to make those chimeric antigen receptor molecules to bind. So what will happen in this case of scenario is, if you generate those CARs and decorate them with all the T cells, then you call them as CAR T cell. Right? This CAR T cell then in case, this CAR T cell that is decorated with the CAR molecule to a specific antigen, it can bind specifically to that receptor, to that antigen that is present on the tumor surface, and then kill that tumor cell. So that is the real simple, the mechanism explained in simple terms. The website that gives you a little more details, I would recommend you to go through that. So the CAR T cells target the cancer. It is a targeted mechanism like lock and key, as I mentioned earlier. So a little closer look. So if you take a closer look, what is happening in this case is the CAR T cell interacts with the antigen positive tumor cell. Look at this, what is happening here. The CAR T cells create a what is called as a granzyme and perforin axis for effective and specific lysis of the cancer cell. So basically, it is like ammunition works like create pores into cancer cell and then kill it by specific lysis. That's one of the actions that is taking place in the, specifically on this CAR T. It also exerts secondarily two other effects which are really good. So what is happening, the CAR T, where the T cells are present and the cancer is present, is a war zone, is a kind of, immunologically, it is a war zone. So in this kind of war zone, if CAR T cell is trying to kill the tumor cell is trying to overcome that kind of killing and want to escape some of those. And the tumor micro, what is called as the stromal cell, which is a tumor kind of microenvironment. So in this kind of thing, CAR T cells derived cytokines can, so the CAR T cell also has a cytokine domain, which sensitizes the tumor stroma. So by sensitizing, it regulates the stromal cells targeting interferon gamma. So it is specifically interferon gamma and interferon alpha receptor, they're all involved with the stromal cells in the tumor microenvironment. So the other aspect that also happens in the antigen negative tumor cell. So there are, so real tumor specific antigen is a myth in the sense that it is also called as tumor associated antigens. Basically, because if you target one receptor or one antigen, the tumor should eradicate, but that is not the case in reality. So there are some antigen negative cells, tumor cells. So if the tumor is the same, it looks the same morphologically, but there are some antigen positive and antigen negative cells. So this kind of antigen positive milieu, the antigen negative fraction can be targeted by CAR T cells by Fas and Fas ligand axis. You all know Fas and Fas ligand axis by the apoptotic mechanisms. So the mechanisms are so multifarious. It causes lysis in the positive tumor cells. It modulates the microenvironment. It also causes lysis of the antigen negative tumor cells. So that is the real advantage with the CAR T cells. So how does this process take place? So CAR T cell treatment, how does this process is initiated? Basically, a patient shows up in the hospital. This is done at hospital or institutions, research institutions, clearly enabled for standard care of therapy. And the T cells are isolated from the patient. And it is through a process called leukapheresis, where immune cells and only T cells are taken up, and rest of the cells are sent back to the patient in a kind of closed system. And these T cells that are taken out from the patient are expanded and engineered in the lab for the chimeric receptor protein to target the cancer cells. All this happens in the laboratory, in the GMP facility. And once it qualifies this, and you create a CAR T, insert a gene for that particular receptor, and create the chimeric receptor molecule that I showed you in the previous slide. And then expand those cells. Again, CAR T cells are expanded. After expanding the cells, they are highly expanded. And then quality control, after quality control and everything, these cells are infused back into the patient. And all this process takes place in around 15 days. The patient comes in on a Monday, and 15 days from then, the patient receives the infusion if everything else is correct. It's a very, very complicated process. It doesn't look as simple as I am telling you. So the engineering part is the most critical part that happens. And this the receptor engineering has evolved over time, over a decade, actually more than a decade. So the first generation, so here, the green color is shown by the antigen recognition domain for the receptor. So this is a receptor part. You can recognize the antigen that is present on the cancer cell. So once it is binds, the signaling domains that are present in signal 1 or signal 2, they transduce the signal to the inside nucleus or inside the cell to take care of the rest. And the third generation also has happened. For these cancer cells to survive better in the tumor microenvironment, they added one more signaling domain. So most of the tumor cells, one of the issues that were faced when this third generation came in, a lot of cells that you have engineered, you have expanded, you have quality control, were infused with the patient. The patient's immune system is killing them. So therefore, in order for them to better, the CAR T cells that you have injected are exhausted. They don't have energy. They are either dead or de-energetic. So to overcome that kind of thing, the next generation, fourth generation or fifth generation CARs have come up. This is created recently by, I don't know, 2016 by NIH. So what they created is this hinge region that we are seeing here, which is shown here. The hinge region is modified. So therefore, this particular one that is shown here is for a B-cell lymphoma. CD19 is a marker for B cells, even for normal cells. But for B-cell lymphoma, the CD19 is a targeted marker, as I mentioned earlier. So this receptor, by improving the hinge region, these CAR T cells reduce the side effects and they stay better for longer in the patient. There are some more developments that are happening. We'll go through as we go through come to the last. So the field, initially was CD19 B-cell myeloma, and there are the FDA approved products target CD19 mainly. So the CD19 is antigen expressed primarily on B cells. So it is CD19 is heavily expressed on B cells and B cell malignancies. So you all know B cell produces immunoglobulin, and that itself is subjected to malignancy here. And followed by the success that I have seen with CD19, there are others like multiple myeloma, which is again you call it as a liquid tumor or blood-related malignancies. And now it is extended to breast cancer, solid tumors, lung cancer, prostate, sarcoma. These are all the targets that are currently in clinical trials. No success yet, right? Only for liquid tumors and for multiple myelomas, or 2 multiple myelomas have been approved. So these are the clinical trials that have approved in United States FDA. And then 4 of them are directly targeting CD19, and 2 of them are B-cell mature antigen, which is also showing which has shown good amenable for cancer treatment. So that CAR T-cell therapy also has been approved for refractory multiple myeloma, for recurring multiple myeloma. So I also see this is what Uday was trying to allude to. So one of the real challenges with this is a very expensive treatment, as I mentioned early on, $450,000. A lot of reagents, GMP process is pretty expensive. So India has taken up some kind of effort by Biocon and few others. And this one particular one that got accepted is a CAR molecule called Next CAR T, which I'm shown here. This is done in consultation, in collaboration with the National Cancer Institute here at NIH, with the collaboration with IIT Bombay, led by Professor Rahul Parwar. And there's also there's a Tata Memorial Center involved, and ImmunoAct, a company involved in this. And I also know a couple of them which are still working on, like Immunil by Siddharth Mukherjee, and Biocon by Kiran Mazumdar. A lot of them are making an effort to get the price reduced to $50,000 instead of $450,000. And this one, as I mentioned, is the whole autologous. The treatment is individualized for the patient. That's where it is all turns out to be very expensive. Even though it is promising, the success rate as of today is only 60%. the 40% of the tumors, liquid tumors, still do not respond. What is happening? So if you look into CAR T cell therapy, mainly it is for hematological tumors, that is blood-related malignancies, and it is autologous, small scale, right? So the patient comes in and you take 50 million cells from them, you expand them to 250 million or something like that, and then put them back. And safety concerns are also very heavy because you can see the side effects that are challenging that are shown. And some of the issues are also associated with this is, you created a CAR, the antigen in the tumor can change its morphology in terms of antigen presentation. So the antigen escape, just a simple nucleotide change can result in a mutation, and the antigen escapes under what is called as immune evasion. So your CAR T cannot recognize it anymore, right? So that is the antigen escape is a huge issue in this. And then the other effect is on target off tumor. I said CD19 is a regular marker that is present on B cells. So therefore, not only the tumor cells, even the regular normal B cells get impacted by CAR T. The CAR T kills those normal cells. The body has to recuperate to generate the necessary normal B cells. So therefore, on target, it is on target, it is killing CD19, but off tumor. So therefore, that's one issue. CAR T cell trafficking. You inject cells, you infuse CAR T cells, and these do not traffic to the tumor. Particularly, if it is a solid tumor, they cannot infiltrate. That is, infiltration is getting into the tumor environment fully. That is not there. And then the other effect that is also seen, immune suppressive microenvironment. The whole cancer is trying to suppress the immunity of the person. Therefore, it can grow. Right? The immune suppressive tumor microenvironment is a huge impediment in the success of CAR Ts, even if there are other small challenges. There are life-threatening activities associated, even though the cells are from the same patient. Because of CAR engineering, because of that, there are life-threatening toxicities, which we will see in the next slide. Modest anti-tumor activity, because of we don't know the exact reason. The anti-tumor activity is very modest. It's only 30 to 40%. There is antigen escape, which I mentioned to you. There is restricted trafficking, limited tumor infiltration, limited to liquid tumors, and solid tumors are yet to be seen. The success is yet to be seen with solid tumors. These are some of the real challenges that are currently being faced. And what are the real toxicities associated with this autologous therapy? So one of the main things that was first tackled is cytokine release syndrome. Once you inject this, infiltrate this cell, infuse these cells, some of the things that is happening in the patients is TNF alpha, interferon gamma, and IL-6 are being released extensively, and that is indicated by high fever, hypotension, hypoxia, terminal organ dysfunction. This all happens very quickly. Like in the first 10 days after infusing the patients, you will see these ones. There are now ways to deal with some of these kind of things, but these are that one has to be cognizant of the cytokine release syndrome. And since the treatment is individualized, it is different from patient to patient. One person experiences cytokine release syndrome, while the other is completely normal without cytokine release syndrome. So the second toxicity associated with this immune effector cell-associated neurotoxicity syndrome, we call it as ICANS. So encephalopathy, right? The whole brain gets expanded, a little bit of expanded. So once you expanded encephalopathy, then there is pressure on the cerebrospinal fluid, and there are a lot of issues coming up of cognitive deficits, seizures, and brain edema. So you have to puncture and take care of the cerebrospinal fluid in some of these patients. So it's a very high neurotoxicity syndromes are associated with some of this one. And this one we talked about on-target off-tumor toxicity. So it is working on the right cell, but that right cell is sitting on a normal cell, right? So therefore, we have to find ways to bypass the normal cell escape from the CAR T. So this results in damage of tissues or organs outside the tumor. So that is also a significant consideration that we have to, significant dimension that we have to consider. So what are the potential solutions for all this? We mentioned autologous. Autologous is low immunogenicity because it is advantageous because it is your own body. You are not going to produce immune against your own cells. And persistence also is reasonably better compared to allogenic, right? So persistence, these are your own cells. Even despite CAR engineering, these cells stay for longer, little bit longer. Disadvantages is complicated logistics. So the logistics is the patient comes into the hospital and in 15 days, a lot of things have to take place very properly to use those cells back into the patient. Variability in starting material. So ages, sex, already these people are showing up with different cancer, different kind of treatments that they have been through for cancer. And the product ultimately that you have made is variable. For simple one thing is the telomeres and T cells slowly disappear. So they age fast and the treatment is pretty expensive. And to overcome this one, allogenic cells is touted. We're still not sure. They decrease the time to treatment basically because the cells are ready in the shelf. When the patient walks in, the patient on day one gets the treatment. These are high quality starting material. If at all we make really good allogenic cells, it is a good starting material because it went through manufacturing, a lot of batches, and then a lot of these batches are QC and very clearly QC'd in and QC'd out. And you have the ability to standardize this product and prescribe it like a drug or like a simple medicine. And because of the scalability and economies of scale, there is a decrease in production cost, and the cost of treatment currently can be reduced drastically if allogenic cells come into picture. But what are the risks associated? Risk is GVHD. GVHD is a process, graft versus host disease, when other cells other than you, cells from any other individual is put into a patient, your graft, your body tries to reject it, right? So there is a graft versus host disease that shows up as rash on the body and all that. We have to find ways to reduce that GVHD. And the risk of alloimmunization. So the alloimmunity is one of the that there is a alloimmune rejection. For that, you have a receptor called the ADR receptor, alloimmune defense receptor, that can be tackled and be worked. I will show you in the next case. And there is decreased persistence. Decreased persistence in the case, because this is allogenic, it is coming from a body other than you, your body rejects it with the cells. So therefore, the persistence of the cells is relatively less. But if you make it in such a way that you can overcome the tumor, whatever is causing the tumor is killed, then the treatment could be in a success factor. So these are complicated things. So some of them is whatever I talked to you is shown in the targets here. We'll see the next in the next slide. How do you make this allo things to work? So how do you come up with the allogenic cell that could be treated in your body as your own cell? Right. So basically, whatever the T cells you're injecting, whatever the CAR Ts you're injecting, the body should feel that it is your own body, right? But which is not. So some of the scientific technologies that are happening as I speak to you is this is called off-the-shelf CAR T cells. Off-the-shelf CAR T cells are allogenic CAR T cells. These are all synonymous terms. So one of the things is you have a TCR. You can knock off that TCR completely. That will take away the rejection from your body. But it has its own downside effects that we'll talk later. And similarly, whether it is alpha beta TCR, the MHC1, which is a immune complex situation, MHC1 can be knocked off to recognize that as your own body. That's another technology that you could use. And then there is another surface antigen called CD52. If the CD52 is knocked off, and the body can still really feel that it is your own body, the cells are coming from your own body. So these are all experiment strategies. Nothing has worked out proven fully yet. So the other one is alloreactivity, activated host cell. The alloreactivity, what I mentioned is if a foreign cell is injected into your body, This is the first thing that responds, allo defense receptor. So you can silence that allo defense receptor ADR through a chemical called 4,1-BB that can bind to it and it can neutralize. These are some of the techniques that are used. And this treatment also is getting really, really close in terms of 4,1-BB. So tumor-associated antigens, I mentioned to you earlier, it is not only tumor-specific, you can come up with a card for tumor-associated antigen, and you can create a card for that, and you can simultaneously post it. So use of pluripotent stem cells. Pluripotent stem cells is called iPSCs. I think many of you know about it, or just Google it. It provides an unlimited source. And if you can create an allogenic model using iPSCs from a really healthy donor, young and adult healthy donor, then the source could be last forever, something like that. And then the co-targeting multiple mechanisms of immune, all this co-target either one or two or three combinations of this. So employing multiple co-stimulatory domains. Remember the NIH receptor that are changes that they made into hinge, that kind of co-stimulatory domains you can create. So CAR ligand targeted vaccines can be created. So, and similarly, in the tough cases, you can give multiple doses of short-lived CAR T in an attempt to preempt the exhaustion. So let's say in 15 days, the cells are getting exhausted, you give one more dose. So maintain 3 or 4 doses when you manufacture the CAR molecule. So these are some of the other strategies that are being followed. And there are other cell types that are in part of adaptive immunity, that are part of innate immunity, so like NK cells or NK cells and NKT cells. They are in the periphery of both of the systems. So therefore, these cells and alpha beta T cell, gamma delta T cell, these are all the sister molecules of T cells that could be made into off-the-shelf allogenic cell therapies. These can be injected into other patients. So the advantage by creating the CAR generation and gene editing and creating the CAR molecules for respective cell types is that you can create off-the-shelf CAR cell product, storage, you can biobank fill and finish, you can do a biobank facility, and you can treat multiple patients from a single donor. So that is the real advantage of this system is from a single donor, you can expand, store, and use the treatment multiple patients. We are not there today, but my fervent hope is in less than 5 to 10 years, we'll be there in that path with all the developments. So this is one slide. The figure is a little heavy, but I will take you through. This is the latest that I have gotten. Their synthetic biology is the approach where you can create a lot of switches for these receptors, as a killing switches. What is a killing switch? Let's say if the tumor is not being erased and it is causing a lot of issues, the CAR T, you have infused CAR T, which is causing issues, you can kill that cell by turning on this, what is called as a killing switch. So that is one possibility. Adapter switches, you can adapt the cells. You have a small molecule switch, chassis cell modification, light operated. You can flash light and you can activate the CAR T cell to kill the tumor cell. So ultrasound, target tumor affected, redirected migration for specific targeting, and Boolean logistic gates. So there are many approaches that are currently going on. The field we are in a fascinating time to take this to a next level. So you can anticipate the emergence of safer, better controllable, better intelligent, and universal CAR T cell therapy in the future. And in all these Boolean logistic gates and all that, artificial intelligence and machine language is heavily used to create those kind of possibilities, right? So your imagination is the limitation to overcome the challenges that are associated. But the success that we have seen in autologous cell therapies is a huge driver for a lot of us academically, scientifically, and industrially to be on this path to improve the healthcare on this. So one of the other kind of things, the conventional manufacturing, it is taking long for the system. There are ways coming up now with simple systems to make it into 1 week 5 to 3 weeks for large scale expansion. So those kind of possibilities are coming up in terms of manufacturing the CAR-T, CAR receptors, whether it is T cells or even for NK cells towards allogeneic. So this is a simple checklist that I have made. I don't want you to go through each and every letter. So this is the workflow process. In an allogenic manner, you isolate the leukocells, expand T cell, and you create a CAR T molecule. You electroporate, you do the gene therapy, you knock out any of these molecules, CD52 or TCR that I talked, and afterwards you expand the cells, you purify, you QC, you freeze and inventory, and it can go to many, many patients. So in each of the steps, these are the checklists that you have to go through, and each checklist has 30 things to, for characterization of the master bank here, you have at least 40 markers to be evaluated simultaneously by FACS, as well as immunohistochemistry, and some of these kind of things that are associated with the QC of this product to go into the patient. So this is a very complicated process. In each of the steps, you have multiple steps to go through, and it's pretty expensive and rigorous. But when done, you are contributing to human health care. That's where the cost is coming from. These are simple terms that I have in the industry, where in a GMP facility that I have worked, how does the process workflow take place? It go through all the steps that I have mentioned to you in the previous slide. And then who is responsible for what? And do a manufacturer supply or a supplier? You may get your reagents from many manufacturers, and how do you audit them? How do you go through them? Right? The CFS is one thing, and whatever the supplier has said, you are not able to reproduce in your thing, and then it is not suitable, right? So there is a kind of checklist that you have to go through. Who will provide who will do all this? And the FDA keeps an eye on all of this, whether it is being done or not, when they come to you for audit once the CAR-Ts are manufactured. So this is a very, very rigorous process. But there are things going on really well, promising things. So a kind of summary, CAR-T cell therapy is a personalized treatment because we are using the patient's own cells, and it has been shown successful for blood cancers, but involves genetically modified T cells. Therefore, it is CAR-T therapy. It's a gene therapy. It is used as a last resort after treatments like chemo and radiation have failed currently because of its expensive nature. Challenge is it is expensive because you have to produce everything for one patient, and very rigorous manufacturing requirements can cause severe side effects and toxicities, as we have seen. Research is ongoing to address a lot of limitations. And future advances, I foresee iPSCs being used as a cell source to generate CAR-Ts and CAR-NKs, and developing CAR-Ts from a healthy donor in an allogenic mode is one of the things that are coming up with the varieties of tests, and creating safer CAR-T cells, fifth generation, using the safety switches that I mentioned, synthetic biology approaches, artificial intelligence approaches that are made to come up with a better ammunition to tackle the cancer, particularly for solid tumors in that. So I think the credits, this is one site that I have extensively used where a lot of information is present, and besides the NCI, National Cancer Institute. And I'll stop at that point and take any questions. Thank you all.
Thank you very much, sir. Your talk was very insightful, and CAR-T cell therapy itself is evidence that there is hope and there is bench to bedside translatability of these innovative cutting-edge technologies. And FABA US chapter highly appreciates your contribution to cancer research, and especially focused on CAR-T cell therapy. And now we can take questions from the participants. Anyone?
Yeah, Deepika, this is Uday. Oh, sorry, go ahead. Somebody else wants. Go ahead, please.
Hi, Mohan.
No, it's fine. I'll just hold
Okay, go
it.
ahead. Yeah, go
Okay.
ahead.
So, Mohan, this is Uday. Brilliantly presented. I think you made a very complicated area, very easy to understand. So thank you so much for that. I'm currently engaged in developing two drugs. One is for breast cancer and the other one is for AML and CML, chronic and my understanding is for AML and CML, CAR-T really does not work. And I've been told many different reasons for it, but what do you think is the real reason? And is there anything on the horizon that you think will fix those issues for specifically AML and CML?
Yeah, thanks, Uday. I think the antigenic variation is huge in AML. That's the big trouble for CAR-T. You know, CAR-T is very specific, right? That might be good in one way and bad in many other angles. Unless you get the right cell in contact, it won't act. So therefore, so that is the real limitation. And you're absolutely right. AML and some of these ones are real is a lot of work to go, a lot of not success. And even in those patients, right? So the ethical and demographical look and feel same patients. One patient is responding, one patient is not responding to this. I think I suggest I didn't go through why some of these trials or some of these things are not fair, not successful. We could have one more session, maybe sometime for the other ones. Why some of the CAR-Ts are failing? Where exactly is the issue that is associated? Carl Jung from University of Pennsylvania has done extensive analysis, and he presented this work last in one of the last conference cancer meetings in Washington D.C. It's amazing to see how the tumor microenvironment in the human body can be so evasive, and it can dance around your antigen. So if it goes in one direction, it can find 10 other directions to escape. So these are some of the issues that are associated with AI, and I can't go in detail in one of them, but I can send you some information.
That'd be great. Thank you so much, Mohan.
What I can add is, Uday, this is Rao from Harvard. AML is a very tricky one. One is, it is in a different category compared to lymphocytes, either B cells or other leukemias. And also, they are in number of tissues because myeloid cells are in number of tissues. So that was another part of it. So now we are doing 3 different CARs against AML at Harvard using myeloid-based receptors. So hopefully in another year or so, we will have a CAR.
Rao, thanks for jumping in. Rao, thanks for jumping in. He's one of the experts from Harvard. Now you have an expert here from no less than a place like Harvard. You should all make use of his expertise to help in this field.
Yep, certainly do. Yeah, thank you so much.
You're welcome.
Mohan, I have a quick question. This is Damodar Reddy.
Yes, Damu.
Excellent talk, Mohan.
Nice seeing
As
you.
always, you made it a very complicated thing, very highly simple. Wonderful presentation.
Thank you.
So the quick question that I have is, what are the limitations of, let's say, this has been evolving, tremendous progress for the past 10, 15 years. I don't do, I'm a biochemist, cancer biologist. I don't do the latest technologies that you follow. It's awesome that you're following it up. What are the limitations? And in this field, as of now, like side effects, tumors that may be arising, and in 10 years from now, where this field will be? Two questions. The limitations, side effects, and tumors because of these therapies in different tumors. The second one is for the solid tumors. The third one is, where do you see the translatability, applicability of this field in the next 10 years?
So you have question after question after question, Damu.
Three questions.
But I'll try to answer. So the first success for this is realizing what limitations you have, right? So the limitations of the cell therapy is very well known, understood, and each of them is being addressed by a group of really talented and intelligent individuals, smart individuals, applying latest and best of the techniques that are available. So that's one thing. The second thing is RNA therapeutics that is happened with COVID cannot be underestimated. So some of those learnings that we have in terms of vaccine, some of those learnings that we have in terms of sustainability or persistence of these CAR Ts that we are injecting have to be used, and the kind of vaccine could be created, a kind of cancer vaccine could be created using those kind of technologies. That's a kind of second approach, I would think. The third one is, the one slide that I showed with a lot of smart switches to clean up, and if the cell is not behaving in the way that we expect, you can kill it, right? So these targeted kind of things and appliance of smart switches, sensors, and artificial technologies that are happening is like you are trying to skin the cat in every possible angle, right? So what comes out of it? I stay positive basically because we were nowhere 10 years ago, and we have now standardized care for liquid cancers. We were nowhere 20 years ago for HER2-based breast cancer treatment. Breast cancer can be completely eradicated if that antigen is HER2, right? So India also has shown really, really well on that. And then in the manufacturing constraints that we have, countries like India and China will top at some day, basically because of the manpower, the intelligence, and the global hub that they have is all facilitating new ways, new avenues of. And China is well known for assembly line, right? I know I may have a lot of criticism on several other things, but assembly line production, you have to give credit to China, right? So some of those kind of technologies, how to manufacture it in a lesser time, how at a lesser cost. So those are the kind of things that are happening, right? Allogenic, even though I showed multiple sites on the T cell to knock off into either 52 or MHC1 or beta-2 microglobin, you can do any of them, but you have to realize how many of them can you do. The reason being, if you knock off all of them, it will be the shape of a dolly. You must all have remembered Dali, how it died. The sheep that was cloned, it died of immune suppression. So if you knock off all this MHC1, CD52, and all those kind of things, the cell is going to die by itself. So you have to optimize what is good, what is bad, how much is you can do on it. But it's evolving science, though, and I stay really, really positive on it.
Okay. Thank you, Mohan.
So can I make one comment on future actually, and a couple of safety issues also?
Please,
So
please.
some of the safety issues are like when you activate these T cells, there may be some insertional mutagenesis, overexpression of target gene, which may lead to oncogenesis. The purpose will be actually nulled because of this. And also if the T cells are activated by viral vectors, they can undergo series of events to generate replication competent lentiviruses that may also give some problem. So for growth in this cell and gene therapy, currently, as Dr. Mohan showed us, there are like 14 approved so far. By 2030, people are expecting at least 100 approvals by FDA, followed by European Union and all. The total is going to predicted to jump at least to 1,000 or so worldwide. So these are some of the projections. Actually, cancer itself will be around 2,000 approvals in next 10-15 years.
Thank you, Prasad. That's a very Rao Prabhala, who is here, can provide how many cars you can come with.
I just want to. Thank you, Mohan. I just want to mention to everybody. This is first time I'm joining. I'm sure some of you are well familiar. I would greatly appreciate if you say where you are from, so that way we will have an idea who you are, not just your name. To ask Dr. Reddy's questions, answers are, is that the limitations with the CAR Ts are predominantly threefold. One is the expense, particularly in US, it's $500,000. So we want to reduce that. And the two is that you have relapses. So all the CAR T, the magic of the CAR T is there's no other therapy in the world that we know of. For the last 50 years, I work with cancer patients. I came into this country 1977. So all the cancer treatments, surgery, radiation, you name it, none of them will yield a response within 2 weeks. CAR T, you will have within a week or 2 weeks. It's a completely gone. So that is the magic of it. So CAR T will, next 2 decades, if you guys remember HIV, how it ran the world, CAR T will run next 2 decades or 3, will be one of the magic bullets in the cancer treatments. The third limitation of it is whatever manipulation we do outside, even though it is expensive, it is coming from a bad immunosuppressive cancer patient. So those cells are not extraordinarily great compared to healthy human being. So that is another limitation. And I agree with Prasad, saying that there are some indications already. People who developed, like Carl June, started almost a decade ago in Philadelphia, that these particularly lentivirus and AAV are still somewhat safe, but retroviral vectors, we know that they integrate, and also they integrate at the promoter regions, whereas lenti and AAV predominantly integrate, we know that, in the intron, which has some regulatory elements, but at least it won't stop the given protein transcription or things like that. So that is slowly will come up. And other cancers will come following CAR T therapy. It hasn't been seen, but Dr. Reddy, we have to wait for that. Okay. The future, you ask the third question, is that what do we see in 10 years, CAR T? In my point of view and the number of people, I think because of the expense and the technology involved to make the CAR T, this is all going to be in 10 years. We're talking about 1930, 2030, 35. We're talking about a box removed from the shelf and give it to the patient, which is allo. So, which is also can be made extraordinarily healthy, taking advantage of all sorts of immune characteristics. So that is going to rise. There are some issues ironing out. There is no allo in the phase 3 clinical trial in US that I am aware of it, or we're not doing it at HMS at Harvard. But we have number of NK cells, gamma delta cells, and we are doing currently, there will be another 4 or 5 CAR Ts approved by the end of this year for liquid cancers. And there are 2 of them are in phase 3 trials, will be approved for solid tumors. So CAR T is going to drive no matter what it is. But the allo will be the future. It will be cheaper. It will be ready to go. There is no waiting. Even at Harvard, patient has to wait for 3 months, even if he is qualified, because in US, it is paid by the Medicare. Most of them are paid by the Medicare, whereas in other countries, it's a different story. So they have to wait, and they have to meet the criteria and the manufacturing process and all that. I hope that that helps you.
Okay. Thank you, Dr. Rao. Yeah. Yeah.
So there is one question on the messages. Also interested, the concept is being considered for autoimmune diseases, where interferon and TNF alpha, IL-6 play a big role. Yes, it is. Autoimmune therapy, parties are being addressed for autoimmune diseases also. We can have a different session on that for autoimmune.
Yeah, actually, we are doing SLE phase 3 trial right now.
Yeah.
So it's an autoimmune disease, so that will be approved in less than a year. So autoimmune diseases are the next come to the cancer for the CAR T, but there is no limit. We are working on infectious diseases. We're talking about cholera. We're talking about HIV. We're talking about Ebola. So all sorts of, any disease is not, there's no exception for CAR T. It's all how we design and how efficacy they are need to be seen, but there are a number of people trying infectious disease and autoimmune disease.
Any specific reason to choose iPSC for future CAR T treatment, sir?
Yeah, I was just getting to
This
that
one.
question. So IPS is an unlimited source. That's number one. And you can manipulate them in the way that you like. Number two. Number three, the allogeneic, for example, a company called Fate Therapeutics is doing the trials right now for NK and CAR NK, some of these kind of things. So the advantage real there is you can get from a real good, healthy, young adult donor cells. And because of the re-engineering, everything has gone back to zero. So therefore, they are better in terms of immunity responses. And you can create an allogenic model by deriving T cells from iPSC. So these are the options from iPSC. Is better than knocking off the TCRs, right? So that is the approach that people have taken, and it is showing reasonably good promise as of now.
And another question from Mr. Sandeep. Can you please emphasize a little bit more about strategies to mitigate T cell exhaustion and applicability of inducible CAR T cells?
Yeah, so I think I replied on the message on the chat box. So one of the things is, at least exhaustion, it is coming from internally within the cell because of the suppression of a transcription factor called FOX. They are working on how to work in FACS. Is pronounced like FOX, F-O-X, is one of the transcription factors that is highly identified, not only in T cells, even in NK cells. That particular transcription factor is highly suppressed, and people think that is what is causing exhaustion. So people are trying to adjust that inside by overexpressing FOX and see how exactly can we sustain that.
Something I can add is that the exhaustion is a very, very complicated issue. Right now, we are doing a single cell sequencing for 100 patients who have gone through CAR T therapy. And what we are seeing, we have two groups of patients. One has almost 3 years, 4 years into CAR T therapy, has maintaining stringent CR, complete response. And there are a group of patients who relapse from anywhere from 6 months to a year. So what are the differences? Of course, there is exhausted cells higher in relapse, but that is not the only story. Exhausted cells are manipulated by a number of mechanisms, one of which is predominantly CD4. CD4 will dictate how the exhausted cells, so there is a paper just came out in Nature Medicine saying that some of these exhausted cells can still kill tumor cells. So how are they killing? Because of the CD4, because CD4 maintains them. And also stopping completely exhaustion is a very difficult task. So what we are trying is, how do we supplement? So you give a CAR T therapy, and then you start with some various sorts of checkpoint inhibitor, a single antibody therapies, or I'm sure Mohan mentioned in the beginning, right now all the approved CAR treatment, there is a bridging therapy to go with it because of the 3 months waiting and lymphodepletion and all that. That bridging therapy is where we can reduce the CAR T therapy. So what we are using right now, there are 4 clinical trials going at Harvard saying that bridging therapy is bispecifics. So there are bispecific antibodies. You use them to make the immune system better, and then you come back and give the CAR, and the exhaustion will be less and the survival will be longer. So that needs to be seen, and those will come out in FDA recommendations in a year, or definitely less than 2 years. So all those factors will play an important role, and exhaustion. I personally feel that we have to allow, because that is a normal mechanism. If you don't allow exhaustions to take place, you will be in trouble in terms of the immunity, autoimmunity, all sorts of things.
Thank you, Rob. There is one question on the chat box, saying that whether nanometals can be used in more efficient and all. That is in a kind of work. And actually, some other things that I believe Rao can add is, during this gene engineering part of the receptor, you have to do transfection. So in this kind of transfection, the delivery of material, the delivery of DNA to the cell becomes really, really crucial. I think the nanometals, lipid nanoparticles, and some of these ones are pretty useful in that to create a better models. It is into the place, but not exactly in the treatment, but in creating the molecule.
Yeah, nanoparticles has been here almost last 2-3 decades. That will be an alternative for the viral infusion creating the CARs. But the only problem is like a tissue specific solid tumors where we have to directly inject these nanoparticles, because nanoparticles will go through immune like a vaccine route. So that takes time. So you can't get a response within a week. But breast cancer and other cancers, you can directly inject into the tissue where the tumor is. Maybe that has, but it still needs a lot of work. So I don't know whether nanotechnology will make a difference, but as I agree with Mohan, maybe it will have a place for the delivery of the CAR T cells, and also add CAR T cells for vaccine effect combining with nanoparticle. But again, that has to be approved, and it is still in phase 1 stage at this stage.
Mr. Vijay Kutala, do you have a question? You can unmute yourself and talk. Mr. Vijay? No response.
He is from Dham Institute of Medical Sciences. Jay?
Yeah.
Vijay?
Hello. Yeah, can you
Yeah.
hear me?
Yeah,
Yeah.
we can hear you.
Yeah.
Thank you. Thanks, Mohan, for an excellent lecture. I really appreciated the most of the things. I have one question that about future of allogeneic CAR T cells, because Dr. Rao rightly said that the autologous definitely have some issues. So what you see, look at the allogeneic in future, because that is easily available.
Yeah, and right now it is in difficult steps, but as I said to Damodar in this question, I stay really positive on it, basically because of 2-3 reasons. The China and India population of cancer patients can become another country, right? The volume is so enormous, number one. And number two, these are the manufacturing technologies that are associated. People know there is a challenge. The challenge means it opens up opportunities. It opens up business opportunity. It opens up scientific opportunity. It opens up everything, right? So therefore, many and smart, intelligent minds work on it and come up with a solution. So and therefore, I would also say with a kind of precaution, right? Allogenic cell therapy is not a natural extension, even though it looks like a natural extension for autologous. Autologous therapies are not going to go away. When you I look at it from a business angle also, right? So it is 2 different customer segments, in my opinion. So therefore, you have the business for autologous, the tools that you require, the process that you require may be completely different. Allogenic is again a process different and scale and reducing the manufacturing complexity, coming to the market quickly, right? So therefore, both the markets will exist, which means the people who invest to work in the area of allogenic need not be bow down. They
Yes.
have still their market for autologous. So therefore, allogenic models is going to go and develop. And I think if we all meet in next 10 years, right, at the same time, we'll be taking a completely different picture of it.
Yeah. Thank you. Thank you very much.
Thanks, Vijay.
Yeah. Thank you.
I have a question.
You can say, maybe you can say 1 or 2 questions and then you can start.
Yeah.
Dr. Mohan, I would like to ask one question. It's a very simple one.
Please.
It's an excellent presentation, and thank you for updating us. And in general, cancer cells are embryonic in nature, and many of them are haptens. And they are, I understand if they are foreign, it will work. But if they are not, and as you mentioned, allogenic, and what are the dangers of developing autoimmune?
Yeah, go ahead, Dr. Suresh.
What are the dangers of the autoimmune developing the autoimmune factors like?
Yeah, is a good point. I see that kind of issue with iPS derived. Now, there were a couple of questions on iPSC, right? So if the cell, let us assume you take iPS cells, you take 100 million iPS cells, and you generate billions of T cells, and you do the CAR engineering on this T cell therapy. So what happens is, in that few billions of T cells you have made, even if one cell for 10 to the power 6, which means one embryonic cell in 1 million differentiated T cell that is present, that can cause tumor. So
Okay.
that's why the FDA puts a kind of heavy emphasis on it. If you are using iPSC, you have to come up with a criteria to show that your final vial that you are infusing into the patient does not contain one iPSC line for 1 million cells. It should be less than that.
Hmm.
So those kind of things are ensured. And the differentiated tumors, right, the embryogenic nature is not a problem.
But percentage-wise, if you see how much they are different and how they behave foreign, like.
So I think it's a very complex, Suresh, to answer that. You know, the issue is the tumors grow slow, fast, right? If you take the fast-growing tumors, the development inside has not happened fully. They are in a rush to divide, and even without cytokinesis, the cell gets formed. So these kind of things form very, very complex issues. But unlike the iPSC, embryonic stem cells, I have not seen tumors posing that kind of another potential for secondary tumors. But only in metastatic that happens.
You already mentioned about the expenses involved in it, but how much confirmatively and how many attempts you will have to make to develop one chimeric antigen receptor?
So for autologous, I think Rao can correct me properly. But the way we have made, at least for Novartis and some of these guys, is for every patient, we make 3 doses. And each dose is per kg body weight, 1 10 to the power 5 million cells. 5 million, 10 to the power 6 million cells, 5 10 to the power million cells, 3 to 5 million cells per kg body weight. You can calculate the math, right? So you create 3 doses, 1 dose to go into the infusion to the patient, 1 dose as a backup, and the other one for FDA verification. In the GMP, verification is a huge process. So you need to store 1 vial stability with all shelf life stability and all that. You have to store it for FDA verification at any point of time. So this is costing $450,000 that Rao was talking about, and some of us were talking about.
What I can add,
Hello.
Dr. Suresh,
Oh.
is that that is where the technology improvements are coming. Right now, we are doing a trial where we use 5,000, 5 million cells. So that will come. The only difficulty is right now is it's very hard to tell after transduction how many are CAR positive, how many are CAR negative, even though you started with the purified T cells or CD4 or CD8, it's very hard to tell after transduction with a lenti or retro how many CAR T cells are there. But now we are coming to grips with it. Once we tell within 2-3 days how many cars are there and purify them, and you can give very low numbers, and that will also subside the CRS, cytokine storm, and the neurotoxicities that is associated with large number. Right now, all the approved cars are given somewhere around 400 to 500 million cells because they are a mixture of both CAR T and non-CAR T. So that technology is up and running, and in a year or two, the cell numbers will come down, and also the manufacturing time right now is like 3 weeks minimum for all the CAR T once you get the blood, and for the infusion time is anywhere from a month to 2 months, and that will shrink to a week. So those two are already in works, and FDA is working hand in hand with the companies and all sorts of things. So I will mention a little bit more.
Yeah, I think there's one more question from Jaya Kaja. In the predictive analysis, are there predictive analysis? To the extent I know, so you need to use 40 to 50 tests to characterize the cells that will be infused. So at least with Novartis and Kite and all that, they have taken a kind of battery of facts analysis that could go simultaneously for the 40 markers. So of the 40 markers, a bunch of them, they have a criteria that it has to be expressed. But if that doesn't meet, the cells are not going to be infused. So that kind of QC assays, predictive analysis is definitely being used. But I don't know, it is not like other kind of disorders where the cell morphology is associated, or the age-related macular degeneration, right? So the retinal pigmented epithelia, they die off. There you can take the shape of the cell and you can do a prediction whether the cell will be working or not. Right? So this cell is not look like, but it has to physically work. So that means the features are so different. And maybe over time, you may come up with some predictive analysis. If you take these cells that with such and such morphology, with such and such features, with such and such growth kinetics, and then with this CAR engineering, you can come up with a machine language algorithm to predict whether that will work or not. I have no idea, Jaya. What thinking?
Hello,
I have
Professor.
laryngitis, so I'm not speaking. But that kind of predictive models, they may exist. I'll take a look. But this is very intriguing, Mohan.
Yeah, if you can take a look at it, and if you want me to look into those, I will be happy to look into, and we will possibly work a.
We have done some
collaboration.
Yeah, we have done some predictive analysis on the literature actually with some lab data with Johnson and Johnson many years ago. But after that, we kind of discontinued the product that we had. So maybe I can pick up on that and then do some checking around.
Please
you.
Please do reach out, and I'll be happy to.
Thanks.
Hello, Professor.
Hi.
Hello, Dr. Mohan. Zankit this side. I'm actually pursuing my PhD in material science in mostly into the nanomaterials. I am actually in a sort of building my own team to have multiple research on these things, basically into the tumors and the therapies. Maybe it is benign or malignant. Consider any of them. Moreover, nowadays it has been observed that around 99%, 90% of the cases come out to be malignant. And since I am from a material science background, I'm sort of building my own team. I'm trying to concentrate more on the nanomaterials because I will be having those facilities at IIT Bombay to synthesize the nanomaterials, the nanoparticles basically. How we can just sort of integrate those nanomaterials into these CAR T cell therapy? The question that I have actually raised in the chat box. So I just could you please just brief us or guide the team what I am building, which is actually not an official team, but it is something like we need some support, like support from delegates like you. If you could just.
Ankit, I know I saw your question. I thought I answered it, but sorry about that. I also mentioned, right? So if you all ask me one thing with the COVID story that Moderna has done, the real success for the COVID-19 vaccine to work comes actually from that nanomaterial, right? Besides the antibody and all those kind of things, those lipid nanoparticles that are in the cocktail of the injections that we all have taken, and for which even some of the side effects are also known. The real success of that is because of the nanoparticles that are associated, lipid nanoparticles that are associated. I took that analogy to say that the nanoparticles that you plan to develop somewhere, whatever is in your process, the nanomaterials will definitely help in the gene engineering part, which I have not discussed extensively in this one because of its clear science with CRISPR-Cas9 and retroviral or AAV or LV. If any of these ones, right, either retroviral or LV or AAV, if they want to deliver the DNA material, you need these transfection agents. So the nanomaterials that we are talking about is one of the more far advanced materials, lipid nanoparticles that form micellase, is one of the real areas that are being used that are currently heavily, you can predict the lipid nanoparticle, how it should be, how it is, in order for get into the nucleic acids into the DNA. So I think that is the area, and there could be some other areas in which I don't know, right? But particularly for delivery, nanomaterials could be heavily used.
So for upcoming some doubts or something, can we approach us like in sort of a meeting or a mail?
Yeah, please reach out to Professor Redana. He has a lot of contacts. Baba will help in all this. And we will be happy to jump in anytime, either me or Dr. Rao or some of us who are little into this field. We'll be happy to help.
Ankit become FABA member.
Sorry, sir.
So you may become a FABA life member in India. Even otherwise,
I can't hear you.
Even otherwise, we will help you.
Okay, sir. So did you ask something else, right? Your first question.
Yeah,
asking you to become a member of FABA India.
Yeah. No, I am not exactly. But I don't know if I get that opportunity to be a member because, see, I didn't have any sort of clinical experiences or anything because
you are a student also, that's okay.
Okay, about the society you mentioned, it is FABA, right?
Right. Yeah, we will send you more details. Leave your email and file number.
Okay.
Divya, you can paste the link in the chat so he can take it.
Yeah, I think Divya will.
I'll just share my email ID. And for more help, if we can reach out to you, it is going to be a great opportunity for all of us. It's like another partner of mine, another two partner of mine is also there in this webinar. One is Sushri Naik, the other one is Namrata Tribedi. So it's like something in India, what we are planning is to do something out of the box, because I am into the material science. We won't be getting the details, like a detailed analysis when it comes to the clinical thing. So in my team, we have members from biotech and biomeds mostly, and also we do have a MBBS doctor who is currently pursuing his MTech in neurology, aspiring to be a neuroscientist. So I
Ankit,
just put,
Ankit, I think we will discuss all these.
Yeah.
Yes,
on
I
the-
okay, okay.
suggest
Yeah,
you
yeah,
get in
yeah.
touch
Just really very
offline.
sorry.
Yeah,
Yeah,
that's
yeah, yeah,
okay.
sure.
Thank you very much, Dr. Mohan, for the talk and for answering all the questions in detail. Now, I request Dr. Rao to do the conclusion summary statement. Dr. Rao is from Harvard Medical School, and he has several decades of experience in immune therapies, microbiology, and reproductive immunology. And he's with us, and it's an honor to have you, sir. Now I request you to do the conclusion.
I think before that, I think I have a request to Redina. Because Redina, for the August meeting that you have, I would request Dr. Rao to give a talk in place of me, because I
Oh,
have done
yeah.
the,
Okay,
because
I will request him.
yeah, I will
Oh,
send official invitation, so that way, for the
thank
next, possibly after August, if you have something else, I'll be happy to do.
Okay, okay.
So thank you, Mohan. It is an excellent lecture and such a complicated topic. You make it people understand, and I thank you. And also, you have enormous industrial expertise. And as we envision, I think you will be helpful to make a GMP materials. I think FABA will benefit a lot. But what I want to say is, for all the members or whoever is in the talk, I think Dr. Redenagaru went a long way, and I don't have to give him what he is and what he has done. I think all of you know. If you don't, you can just Google it. It will be everywhere. He has done with a humility and an ambition and passion, particularly. That is lack in the profession, particularly in Indians and particularly in Telugu people, because I have been in this country. Here, we have more caste, we have more political problems, all sorts of things. That's why I don't go to any association. Just imagine, if a Punjab has a one association in US, Telugu people have a 24 associations. That tells you how we fight and how we don't help. Whereas him to see this vision and create something that will flourish even after he is gone. That has to be in that mind frame. See, Steve Jobs is gone. Is Apple died? No. So that is what it is. If it is built in a structural fashion and built with a passion, the money and the fame, those are automatic. Those will come. So I thank you from bottom of my heart to Redenagaru, and I hope he will continue to provide help where people who have his type of ambition and want to deliver. Okay? And in terms of the what Mohan's talk is an outstanding, and I at least want to mention, I know CAR-T since last 2 decades, and Carl June is from Harvard, and who is considered as father of CAR-T in Philadelphia, and we work hand in hand with the Philadelphia group, and I know A to Z, how to manufacture, how to scale up, and particularly in the part of a lot of questions came today, what are the problems? So we know that it's a magic bullet. You will get a response in 2 weeks. The limitations are, how do we make it affordable? How, even though the cost is $50,000, how do we make it affordable in India? How do we make it faster, not waiting for 3 weeks? And how do we use it for, there are 200 cancers, not just liquid cancers or leukemias or lymphomas or lung cancer or stuff like that. There are a number of cancers. And also, CAR-T has a field of infectious diseases and autoimmune diseases, SLE, rheumatoid arthritis, diabetes, you name it. And particularly, there are cancers like neuroblastoma. The last person, Ankit, was asking, I give him a direction. How do we deliver with nanoparticles to glioblastoma? Whereas you can directly inject with a nanoparticle and remove the tumor very quickly. So those type of things are the ones. And the last one, if Reddy Nagaru allows me, I want to make a small pitch to the FABA US. And my goal is to make a manufacturing facility in India because I'm very passionate, because my mother died with COVID in 21, told us or instilled in us saying that if you don't help to the community where you come, no matter what you made, what you learn is all waste in her books. My mother is a failed 7th grader. So on that note, I want to make it in Guntur. That's where I am from. So I'm building a CAR-T facility and GMP facility, hopefully by end of this year. And the problems are, I give you 2 examples. One is when I first came and teach medical students, I give one example. There is a fish tank exhibition and everybody brings their fish tanks with covers and everything. This Indian guy brings a fish tank, there is no cover. And the judges will, hey, your fish will jump out. And that guy will sell, don't worry about it. You know, our Indian blood contains, if a one fish is jumping, all the other fishes pull him down because we won't allow or we won't render the helping hand. That's in our blood. That has to go. That has to go. And hope that the other one is all the ants can stop the elephant to move. So there is a little help. It's not that, because in my last trip is where I happens to meet Reddy Nagaru, though I know him last 20 years and what he has done, and he gave me a helping hand. And I met all the big shots in Hyderabad and all other places in Andhra Pradesh. Nobody, I'm just telling you, it's not a complaint. Nobody even gave an encouraging hand. So that tells you how difficult it is, passionate, knowledgeable, skilled, unable to do get help. So for this manufacturing facility, GMP facility need 10 crores. So whoever want to participate, more than happy to connect with you. And the only thing I can tell you is in another year or so, I'm going to make 10 to 20 cars in Guntur and help like Reddy Nagaru and Mohan and lots of others. And when that day comes, people, yes,
for generous compliments. And I am sure, I think if one is determined to build a manufacturing facility, definitely help will come and FABA will be the first one to help you. And here I request Dr. Pollani Sheshagiri is here in this. He is the one, in fact, providing all the kind of support for the startup ecosystem in the country. Dr. Sheshagiri, would you like to just say a few words?
He was silent. I didn't see him. Hi, Shashgiri.
So he is the one connected with the Indian biotech ecosystem, either in the Department of Biotechnology or Department of Science and Technology or Indian Council of Medical Research. He is the one, in fact, supporting a number of startup companies. Yeah, Dr. Shesh.
Thank you, Dr. Reddanna. It's really a great pleasure to listen to this lecture of Mohan, one of my associates, long-term associates. Hi, Mohan. Your lecture was very nice. Lucid is exactly what is required to open up the number of intriguing questions, the pros and cons. I really enjoyed. And like Dr. Redana mentioned, if I can be facilitatory and helpful in any way, please approach me and I can share my email and I will just help in the way that is possible.
Thank you. Thank you, Chef.
Thank you.
Was from Indian Institute of Science Biochemistry Department. In fact, he is also visiting US. Right now, he is in Fremont area. Will be sometime in this week, in the next week.
Yes. Thanks, Redana.
Yeah. You could probably conclude other things.
Yeah, thank you all. In the month of August, from August 2nd to 16th, we have the Drug Discovery and Development Workshop. We'll be sharing the brochure with you shortly after this meeting. Stay tuned and connect with FABA India and FABA US chapter for more updates and be part of our community and help us disseminate good science across the US and across the globe. And thank you everyone for taking time to attend this webinar, and your presence is much appreciated, and we will see you for more in the upcoming several months. Thank you very much.
We have the next webinar. Announce it, please. Next webinar.
Yeah, we have a webinar coming up on July 6th on plant-based monoclonal antibodies. It's from a company in Thailand. So we'll disseminate more information about that webinar in the group, in the WhatsApp group and through your emails. Thank you.
Yeah. Deepika, you also introduced Dr. Sai Giridhar, who is...
Yes. Yeah, Dr. Sai Giridhar is a transformative health coach, and he's a wellness coach and retreat specialist with a background doctorate in clinical biochemistry. He is with us on this session. Dr. Sai Giridhar has several workshops and holistic health approaches for transformative life journeys. And if he wishes, Dr. Sai Giridhar, if you could say a few words about your...
Yeah, hello everybody. Thank you so much for this opportunity. I had done my PhD in clinical biochemistry. Can you hear me?
Yes,
yes, we can hear you. Go ahead, please. Thank you.
Thank you so much. I had done my PhD in clinical biochemistry in early detection of heart disease. I was privileged to be associated with a cardiologist from Cleveland Clinic and also a diabetologist, Dr. Sanjeevi Garu from Karolinska Institute, who guided the project. And after the successful completion of it, I was inspired to take up wellness from a very different perspective. So the last 6 years of my research has been on human wellness. And today I combine a unique model that is a confluence of scientific research, ancient Indian knowledge systems, and human values into a wellness program. So I am a certified health and wellness coach, also a transformational life coach. I conduct one-on-one sessions and also retreats, especially for underserviced professionals, because all of us as scientists, we know the number of hours we put into the work. There are many such doctors, lawyers, and various other professionals who work very hard to serve the society, but they themselves are underserviced. And I bring in my expertise to help them, develop their own wellness. So that's been my domain of work for the last few years. Thank you.
In fact, Sai is visiting US. Right now, he is in Raleigh area and will be coming to New York, I think, Mohan, somewhere around, I think.
Yeah. Sai Giridhar, please let us know when you are here inside. We'll be happy to meet.
Sure, sir. Thank you so much. Thank you for this wonderful presentation. I was really thrilled to know about the frontiers in this area. Yeah. Thank you.
Dr. Saigaru, so on the way to Mohan's place, so I'm in Baltimore, so I can meet you, like, in case if you're driving from Raleigh to New York, if you're flying, that's okay.
I'm flying to Newark, but I may
Okay.
travel to Baltimore nevertheless because Dr. Govindrav Garu, I'm not sure if you're familiar with him.
Not sure. Huh?
Yeah, so he's into biosensors
Okay.
in the Maryland University. So I may have a meeting with him as well. He has been helping us since our student days back in Andhra Pradesh, Puttaparthi. As the Satya Sai
Okay.
University is where I come from. Yes.
Good. So definitely I'll meet you when you come here, sir.
Sure. Thank you so much.
Thank you.
Thank you very much, Dr. Sai.
Thank you.
Bye. Thank you, Mohan. It was again,
Yeah,
excellent from you. We know right from the student days, Mohan and myself studied at SV University, Tirupati, MSc Zoology, and then PhD also from the same place. And we also together worked at University of Hyderabad. And then anyway, he left us and came to US. And worked
All
in
destiny, right?
institution.
Destiny. Yeah.
All we don't design.
You're forgetting one more person.
Damodar Reddy, sir. He's also.
He is also.
And Prasad is also there, right?
Yeah.
It's nice meeting Srinivas. Wonderful job, everybody. Thank you.
Yes.
This will
Thank
continue.
you all.
Yeah. Thank you, everyone.
Thank you,
We'll
everyone.
see you in the next webinar.
Sure.
Thank you.
Thank you so much.
Bye.
Thank you so much.
Bye,
Have a great
everyone.
weekend. Bye.
Yes.
Bye.
Have a good weekend.
You too.